workout.
Everything we're talking about in this chapter, we're talking about all the processes that are happening inside of the cells, whether it's a bacterial cell, whether it's a eukaryotic cell, it's all the different processes that are happening. As we break things down and build things up, we have catabolism, which are the reactions of breaking things down, and anabolism, which are the reactions that are building larger molecules. One of the things that we are breaking down and building up and also using to help break things down and build things up are proteins. So just some background review of proteins. Proteins are made up of amino acids. There are 20 different amino acids, although some prokaryotic cells do actually have 22 different amino acids, but humans have 20. All 20 amino acids have the exact same basic shape. We'll look at these a little bit in lab. Something I can point with. So they all have a central carbon. They all have an amino group. They all have a carboxyl group. They all have a single little hydrogen. And the only thing that changes between the amino acids is kind of that tealish colored, what they call as an R group. So every amino acid looks about the same, except that one tiny little change on the R group. I was going to see if I can click through these too. Hope this doesn't mess it up. When we string amino acids together, the bond that holds them together is called a peptide bond. So when we put these two amino acids together, the bond that holds them together is called a peptide bond. So now we just call something that's not two of them together a dipeptide. And then proteins are made up of amino acids, and it's never just two different amino acids. There's always going to be a huge, long sequence of amino acids. And they will fold, they'll form these cool little loops, and it will determine their structure, which then also is coordinated to whatever their function is. Enzymes are proteins, which is why I do a quick review of proteins. There are lots of types of proteins out there. I mean, some are used for building things. Well, enzymes are proteins, but they have a very unique function to them. And their unique function is that they do increase the likelihood of a reaction happening. So kind of like, what are enzymes? Enzymes are organic catalysts. So they're organic, they're carbon-based. And they're a catalyst, meaning the next part. They increase the likelihood of a reaction happening. If there are enzymes around, it is more likely that reactions will happen. And with the millions of reactions that are happening in every cell all the time, all day long, every minute, we need these enzymes or certain reactions aren't going to happen. We're not going to be able to break things down or build things up. Now, how they help. So this is showing if we have some reactants, their job is to become some products, whatever they're going to do. The reactants are going to become products. It looks like we're going to take this AB structure, break it down into the products of A and B, whatever they are. Well, to get any reaction going, anything, we do have to put energy in. It's called activation energy. Just as an example, if I have a match and I'm holding a match, does the match spontaneously start? I can't just look at it and be like, go, light up. I have to put a little bit of effort, a little bit of energy, and strike it on the side of the box or the side of the little matchbook. To get any reaction started, you have to put some little bit of energy in. That's activation energy. You're activating what's going to happen. And this is to show, before these reactants can do anything, I have to put this much energy in for them to then eventually become products. But enzymes are around. They lower how much energy is needed. Instead of this much energy needed for them to become the products, only this much is needed. And if you don't need as much energy to go in to start a reaction off, it's more likely going to happen. You probably all lit a match at some point and you're just like, oh my God, this thing is never going to light. I'm like, maybe it was missing something on the match. It was missing the enzyme. But if you've got enzymes around, maybe you only need to just touch that match right to the box. It's more likely to happen. Less energy is needed. Now, I found this from a different textbook, and I love it because my reactant frogs want to become product frogs. And if there are no enzymes around, they need a lot of energy to be able to jump over that wall to become a product, which means some of them might not be able to do it. There's just not enough energy. They don't have enough energy to be able to become a product. However, if there's enzymes present, that wall is a lot smaller, and it is more likely they will be able to jump over it and become a product. So enzymes lower that amount of energy that's needed to get that reaction going, so it's much more likely to happen. So enzymes, as I said before, are proteins. So enzymes are proteins, but not all proteins are enzymes. They're just an example of a protein molecule. And every enzyme, and you'll notice it in different textbook pictures, enzymes usually are represented as big blue or big purple blobs. Every textbook I've ever seen. So enzymes all have what's known as an active site, where stuff's happening. And it's that active site that's going to bond to a substrate. That's the line. So the active site is where that substrate is going to bind. And enzymes are very specific for what substrate can bind to it. So this enzyme is only going to allow this weird-shaped yellow substrate to attach to it. Now, if you notice, does this look like it fits perfectly in there? What ends up happening is that a substrate, it is specific for this enzyme, when it binds to the active site, it forms a more perfect fit. And so it will look a little bit more like that. But it is still specific. Nothing else can bind there. But right away, it's never a 100% perfect fit. But once it fits in there, it becomes a little more perfect fit. Enzymes are usually named for the substrate that can fit. So an example is probably on my next page. I'll just wait. Or on my next slide. They are named. Most enzymes, just to even know that it's an enzyme, their names end in ACE. And that's a huge red clue. It's an enzyme. And the name of the enzyme is then generally named for the substrate. For example, lactase is an enzyme that binds to lactose. So they're just names for their substrate. This, never had to memorize anything like this. I need my pointer. I'm too short for even this slide. Here's the enzyme, again, big blue blob. And its job is to attach into its active site this substrate called fructose 1,6-biphosphate. And because this enzyme is only going to fit this substrate, the fructose 1,6-biphosphate, the name of the enzyme is fructose 1,6-biphosphate aldolute, named for the substrate that it fits. Well, it's not a 100% perfect fit until it gets in there, then it becomes a more perfect fit. The job of this enzyme is to break apart the sugar. So that we can see it's breaking apart the sugar, and then the enzyme goes right back to normal. So kind of for my fill in the blank part, enzymes are not used up. Enzymes are not used up or changed way whatsoever. It just keeps going in the cycle. Keeps waiting for another fructose molecule. They are specific. They are specific for one substrate. Drive a reaction one way because they are a catalyst. So the enzymes, again, they're not going away, unless you would like to change it somehow, but they're not going away. They just keep finding more of that fructose molecule and keep splitting it up, and then they go find another one and keep splitting it up, and that's all they keep doing. They're not used up. They don't get changed whatsoever. Here are some things that could affect how well enzymes actually work. Temperature can affect how enzymes work. That's one of my next slides. Yep. Temperature can affect how enzymes work, how good they are. The pH of the environment can affect how well they work. How much enzyme is present versus how much substrate is present could affect how fast all of this is happening. And if there are any inhibitors around, that can also affect how enzymes work. And these are like the next several slides. So this is just showing that different enzymes work best at different temperatures. Most of the enzymes found in us, because our bodies are 37 degrees Celsius, the enzymes work best at about 37 degrees Celsius. So this is showing the optimal enzyme activity is right around that 37 degrees Celsius. So optimal temperature for human enzyme, well, it looks a little higher than that, but either way, it's going to work just fine at body temperature. However, different organisms have different optimal temperatures that they work best at. There are organisms, little prokaryotic cells that live in hot springs. Well, if you try to put them in our body is freezing cold for them. They want it to be like 70 degrees Celsius or higher. It's not quite boiling, but getting pretty close. That they have a different optimal temperature. So even different bacteria have different optimal temperatures. Generally, the ones that infect us have the same optimal temperature as us. Different enzymes have different optimal pHs. Our bodies are pretty neutral pH, depending where they are. And so most of the enzymes that do different reactions and the bacteria that live in different areas of the body, their pH is pretty neutral that they're going to work best at. And then even how much substrate there is compared to how much enzyme activity there is. So as the substrate concentration increases, at some point, the enzymes can't do any more. And so if you start to increase the number of substrates, I'm going to go back to this picture. 10 of these little enzymes in the body. And I have, 5 of this substrate. Well, if I started to increase more of the sugar, well, I've got more enzymes. The more sugars, the more enzymes can work. But at some point, I can only keep increasing this substrate so much because I still have the same amount of enzyme. So if I have 10 enzymes and 5 substrates, I could increase the substrate up to about 10. And then at that point, all the enzymes are busy. That it does not matter how much more substrate you add, there's only so much enzyme present. And so that's when it hits what's known as the saturation point. That you can start to add more substrate and you're going to get more enzyme activity until you hit a point where all of the enzymes are busy. You can only have so much enzyme activity. And then there is something known as inhibitors that can come along and change how well enzymes are going to work. And there's 2 different types of inhibitors. There's competitive inhibition and non-competitive inhibition. Some of these pictures are from different books. Just like books have different pictures and some people like certain ones. So competitive inhibition. So here's your big enzyme, big blue blob, and here's your active site. And this is the substrate that's supposed to fit in this little active site right here. However, there are some inhibitors around that can fit in part of that active site. And if that inhibitor is sitting in the active site, can the substrate fit in there now? Nope. It's already like, oh, someone else beat me to it. And so it's an inhibitor because it inhibits the work of the enzyme. And it's called competitive inhibition is because they're both competing for the active site. And so if inhibitors around, we don't get any product because the substrate can't sit there and it can't do its job. Again, different pictures. If this is supposed to fit in the active site, but this thing comes along and fits in first, so the substrate can't do anything. It can't fit in the active site. Actually, I can't remember which one is out of your edition of the textbook. This is the normal substrate that's supposed to fit in there, but if this inhibitor gets there first, the normal substrate can't bind, it can't do anything. Again, it's called competitive because they're just competing for the same active site. But then there's also something called non-competitive inhibition. Still an inhibitor, but it's not competing for the active site. So if we still have that same big blue blob enzyme, here is the active site that the substrate's supposed to fit in. But on enzymes, they do have another site that stuff can bind to. It's called an allosteric site, right there. And some inhibitors can fit into that other little spot. And if they bind to that other little spot on the enzyme, this enzyme starts to change shape just a little bit. And on this, the shape, it flipped over. I'm going to go back. This is what it originally looked like, and the substrate could easily fit in there. But that other inhibitor fit in that allosteric site, that alternate site. It changes the shape just a little bit. Because it changed that shape right there by the substrate, the active site, can the substrate fit in here anymore? No, it just kind of flipped it over and kind of closed a little door on it so it can't fit in there. And so the same result is there. You're still not going to get any reaction occurring. The substrate can't bind. You're not going to get any products. It's just called non-competitive because they're not competing for the active site because it binds somewhere else. Same end result, though. This is just showing here's the active site, but things can bind in other spots. And they change the shape of the active site, stuff can't bind. So you need another spot, and the substrate can't bind. Now, what can happen is sometimes the product that these enzymes make actually become inhibitors. So this is a normal, they're called like pathways. If you have an initial substrate going into this enzyme, again, big blobs, they go into the active site of this enzyme is going to take little orange circles and it's going to spit out little yellow triangles. So this enzyme's like, well, great, I love little yellow triangles, we'll fit in there. And I'm going to take it, I'm going to spit out little red squares. And this enzyme's like, awesome, I love little red squares. It's going to take the red square and it's going to spit out little green ovals. And it does this through a whole series of process because the body, or the cell, if all you are is a cell, needs this, whatever it is. It needs this final end product, and it uses several enzymes to eventually make the final end product. But what can happen sometimes is that end product can actually become an inhibitor. And it can go up to one of those enzymes, it can bind to that enzyme. It's binding on that, it's a non-competitive. But I'm like, it's binding in that little allosteric site. It changes the shape of that active site just a little bit so that the little orange circle can't go in and bind anymore. And if the little orange circle can't bind, well, then you're not going to get yellow triangles, you're not going to get red squares, and you're not going to get any more of that initial end product. Those pathways are called biochemical pathways. It's just a pathway to get your final end product. This on purpose so that when a cell makes what it needs to make, it stops making it after that. Like, hey, I just need 10 molecules of little green ovals, and then I don't need to make any more. Like, you only want to make exactly what you want to make, and so that product can then become an inhibitor so that the cell doesn't waste any more time making more little green ovals if it doesn't need any more. So it helps regulate the cell to make what it needs to make, and then just stop. Cells are lazy, efficient at what they do. Now, this is where the majority of my PowerPoint comes from, is carbohydrate catabolism. So I talked about catabolic processes last time because there was anabolic processes and catabolic processes. And what's happening in catabolic processes? Something's building and something's breaking. Catabolic breaking or building? They break things. So in catabolic processes, stuff is breaking down. And so in carbohydrate catabolism, it really truly just means the breaking down of a carbohydrate. Anything you see the word cat, you're like, oh, breaking. Cats break things. Now, when we break anything, we break a bond. And when you break a bond, it releases energy. And when you release energy. And you're like, well, okay, release energy. What is this energy? A lot of that energy that gets released is energy that's in electrons. It's like review of last class. And when you lose electrons, this is my oil rig. When you oxidize, it is drops. So when you break a bond, you lose electrons. That's oxidized. From last time. So many organisms oxidize carbohydrates. Which again is in a fancy way that we are breaking bonds to release or lose electrons. It's that energy in the electrons that is then used to build stuff up in anabolic pathways. You break stuff down, gets energy, build stuff up. That's why we eat. We digest the food, we break it all down, we release all the energy that's in that food we got done eating, and we use that energy to build whatever our cells need. Now, glucose is the most common carbohydrate that's used, and so that's what we're going to focus on. And it's catabolized by two main processes, cellular respiration and fermentation. And we're going to focus mostly on cellular respiration. Cellular respiration involves an aerobic part to it, but it can also be done anaerobically as well. It is more efficient aerobically. Fermentation, breakdown of glucose, without any oxygen present whatsoever. These are my next, I don't know how many slides. We're going to break it down, and I'm going to draw stuff. A lot of stuff going on this slide, and I'm going to break it down piece by piece. So when we break apart, I wanted my pointer. When we break apart glucose, so on my image, we have our glucose molecule. It goes through several steps. If we have cellular respiration, which can happen with air, with oxygen, and it can happen without oxygen, and it can happen both of those as well. A glucose molecule is going to go through glycolysis, and then it can go into the Krebs cycle, and then it can go into the electron transport chain. That's going to be kind of your basic understanding of cellular respiration. Now, advanced A&P, did you do it this yet? Right, because you're in advanced. Are you in advanced? Yeah, have you gone through this? No, you will. Because again, they're not about the same time. Gonna through fermentation. Still gonna go through glycolysis. It goes into the fermentation orange side of my diagram. But we're gonna focus first on taking glucose and going through all of this down through the electron transport chain. I did give you that diagram, that little table to fill in. I'll try to make sure you guys get something filled in. If I miss something, yell at me. And no, am I going to make you ever memorize these? Nope. You do not have to memorize any of these big diagrams. The big thing to know is really what's going in to these processes and what do we get out of the processes. If you're like, this looks super cool, I would love to memorize this. Not at Western. We don't have a specific cell biology class. At UWL, they actually truly just have cell biology, an entire semester, all about the cells. Memorize this all you want. But we're going to start, and I'm going to try to draw some of the basic stuff going through. We're going to start with our glucose molecule up there. Now, do you remember from prior classes what the formula for glucose is? Yeah, what is? Yes? 6. So C6H12O6. Carbohydrates, in all carbohydrates, always have a ratio of 1 to 2 to 1. And glucose is a carbohydrate. And so it has a ratio of 1 to 2 to 1, or 6 to 12 to 6. Now, as I break apart glucose, I'm going to focus mainly on following my carbons. So I have 6 carbons, C6, And up on the very top up there, they draw 6 little black circles with a little C in there for representing the 6 carbons. I'm not going to put a C in the middle of mine. Now, I have students, if you want, you can draw what I'm drawing on the board. Use my screen, draw across my board. But the back side of that worksheet, it's great. It's blank white paper if you want. But if you are going to draw it, like, ooh, this way with the paper, not the other way. Unless you draw small. So in glycolysis, anything that has that ending lysis just means to break apart. Glycolysis truly just means breaking apart sugar. Now, I'm going to start with my 6-carbon glucose molecule way at the top. And my final end result, after all of that, is going to be 2 3-carbon molecules. So that 6-carbon molecule is going to go through some different stuff. I'm going to break down. But in the end result, I'm going to get molecules, and they each have 3 carbons. So we get what's 2 pyruvic molecules. Pyruvic acid is also called pyruvate, just because I think you see it in some of my diagrams. Exact same thing, just different names. I lost any carbons at this point? No, I still have all 6 of my carbons. What I've done so far is break it down. I don't have one big molecule of carbons. I have 2 smaller molecules of carbons. Now, before you have any reaction occurring, what do you need to start that reaction off? There's usually one present, and that lowers what that's needed. To light my match, what do I need to put in? Energy. So to get even this reaction started, because glucose is not going to just spontaneously break down, I need to put some energy in. So at the top up here, arrows, if you can see, I have this molecule of ATP is going in. When it breaks down, that's where you get ADP. But I have 2 molecules of ATP that go in to start this reaction off. And I was going to be like super colorful today. Do not have all the colors I wanted. 2 ATP go in just to get this thing started. Arrows of stuff coming off some of these. What am I getting out of some of these reactions? What's one thing that's coming out of a reaction? And they try to color it and make it all bubbly too. It is, only because I'm going to talk about it when I get to fermentation. Anytime 2 ATP go in, you get ADP. What else do we get out? Little arrows coming out. Look at the sides. And I get many. 2 of them. Now this is from last class. NADH, because I didn't care that you knew what it all stood for, but what is NADH? Why did we care about it? Look back in your PowerPoint. There's NADH, NADPH, and FADH2 were 3 common something. You guys see that? Carrier. Yes, NADH is one of the 3 common electron carriers. And I care when I break down glucose about electron carriers because electrons carry energy. Something else. We do get a couple waters. This is where I wish I had colors because I would have like a color for every single thing. We get a couple waters out as well. Care about what is going in and what is coming out. So filling for the first, where does this happen? Where does glycolysis happen in the cell? This happens in the cytoplasm. So this just happens in the fluidy portion of the cell. For what goes in, we have one glucose, one single glucose molecule, and 2 ATP have to go in, because that's our activation energy. Oh, we missed a couple things that also came out. What also comes out? Big bubbly thing. Near the bottom. Many though. Not just 2. We get 2 out here and we get another 2 out there. So in the next box, we already got what goes in, 1 glucose, 2 ATP. What comes out, at the very bottom, what comes out is 2 pyruvic acid. What also comes out, we have 4 ATP, waters, and 2 NADHs. You get a lot of stuff coming out. Not too concerned yet about the ADP, because it's just the breakdown of ATP, but it will come into place when we get into fermentation. Leave it up there. We get a lot out. We got 2 NADHs, 2 waters, 4 ATP, and 2 pyruvic acids. So the next box that asks for energy produced, that's always going to be in the form of ATP. I made 4 ATP, but I also put 2 ATP in. So how many have I actually gained? Only 2. So this whole process, that's a lot of stuff going on, gave me 2 ATP. The net ATP that I've gained. Anything else important? The biggest thing with glycolysis, glucose turning into 2 pyruvic acids. Again, lots of things happening. Lots of video is. This glycolysis process, for anything else important, is part of cellular respiration, also part of fermentation. So glycolysis is part of cellular respiration and fermentation. Otherwise, it splits up after that. There are completely different processes going on. The very bottom part is a part of fermentation, is a part of aerobic respiration, or is a part of anaerobic respiration. All of the above. It's a part of all of it. One process. What happens to those two pyruvic acids? So pyruvic acid, or pyruvate, again, continually starts to get broken down, and then it's going to go through three more things. It's going to make something called acetyl-CoA. In my next slides, it's going to go into the Krebs cycle, and then it's going to go into the electron transport chain. But we're going to start first is that pyruvic acid that came out is going to get made into a molecule called acetyl-CoA. So pyruvic acid, again, this is why it's also called pyruvate, folks. Pyruvic acid, or pyruvate, is going to go in. That's because it's now my new beginning reaction. And it's going to get broken down. And I'm only going to follow this top one. Just know whatever I do with this, also happening down here, but I'm going to make my board look less confusing. So it's going to get broken down, a series of things happening, and it's going to get broken down into a 2-carbon molecule called acetyl-CoA. Sometimes called acetyl-coenzyme, acetyl-CoA. There's not a lot of other things going into this step, less arrows of this, but there are some things that we get out. What's one thing we get out of taking pyruvic acid and making acetyl-CoA? What's one thing that we get out? You see arrows going out. NADH. At least I used a different color. We get an NADH. Now, NADH happening here. Just note into this one too. So how many NADHs do I get so far out? 2. Because again, it's one molecule of glucose. I get an NADH here. I get an NADH here. So I gain 2 NADHs. And what else do I get? It also is coming out. CO2. And that becomes important because I went from a 3-carbon molecule to a 2-carbon molecule, and that carbon didn't spontaneously disappear. It left as the C in CO2. This is why we breathe out carbon dioxide. Not as much happening in this. Though, where does this occur? Cytoplasm of the cell. It's still happening in that fluid. For what goes in to pyruvic acid? I'm only going to follow and draw out one, but there are 2 pyruvic acid, and this is happening to both of them. So 2 pyruvic acid go in. That's it. Out of the making of the acetyl-CoA, and again, it's happening in both of my pyruvic acid. What comes out? I'm going to get 2 NADHs. One here that I drew, and one down here that I'm just not going to draw, just to try to make my board look simpler. I'm also going to get out 2 CO2s. I'm going to get out 1 from this process and 1 from this other one that's up here that I'm just not drawing. So I get 2 NADHs out, I get 2 CO2s. And I get 2 acetyl-CoAs, 1 here and the 1 down here that I'm not drawing. Things out. 2 is, well, yes. Types of things and 2 of each of them. But I get 2 acetyl-CoAs, 2 carbon dioxides, 2 NADHs. And again, why do I care about NADH? What is NADH? Carrier, and I care about electrons. Do we get any energy out? Do we get any ATP? Often, nada, 0, whatever you want to put in there, I don't care. It's your table, I'm not collecting it. For the anything other important, at this point, it's going to go to the Krebs cycle. So something along the lines of it's going to go straight to the Krebs cycle, or acetyl-CoA is going to go into the Krebs cycle. Once we've made acetyl-CoA, we are going to the Krebs cycle. Is a part of fermentation aerobic or anaerobic respiration? This is where it gets a little different between what you learn in advanced A&P. It could be part of aerobic or anaerobic, depending on the organism. There are anaerobic bacteria that will do this anaerobically. Aerobic organisms will do it aerobically. And are aerobically and will only do this aerobically. So in advanced A&P, you'll be taught that this is all part of aerobic respiration. It is, but just know microbes can do this anaerobically. We can't. Either or, aerobic or anaerobic, depending on the organism. Cycle. Again, I don't care about all those steps. I'm mostly focusing on what is going out and what is coming in. So that acetyl-CoA go in. It's way at the top up there. Acetyl-CoA is going to go in. And this 2-carbon molecule is going to meet up with a 4-carbon molecule. And 2 plus 4 equals... What's the name? I don't care about it, but I do refer to it. A 2-carbon acetyl-CoA is going to mix up with this 4-carbon oxaloacetate. The long version is oxaloacetic acid. And the 2 is a 6. The 2 plus 4 will give us a 6-carbon molecule. I don't care about the shape. Oh my God, we're right back to where we were. Yeah, but it's not the same structure. But this is called citric acid. The only ones I'm writing up are the ones I referred to, because sometimes the Krebs cycle is also now called the citric acid cycle, and it's because the 2 plus the 4 gave us this new molecule. Well, the citric acid cycle is going to have lots of things that I don't care about. Again, if you love it, take sell by it, you will. I only care about things that we get out of all of these steps, because at some point it is a cycle. We're going to do lots of things to that citric acid, and it will go right back to that 4-carbon oxaloacetate. But I care about what are some things we get out. What's one thing that we get out? ADH. And how many throughout the whole Krebs cycle? 3. We get 1 here, 1 there, and 1 way up there. So out of this one Krebs cycle, just one Krebs cycle, oops, I'm not going to draw them all separately. I'm going to get 3 NADHs. What else do I? How many? 2. I get 1 there, 1 there. Again, I care about the CO2 only because this is accounting for my carbon. How many? What about FADH2? One of those 3 top electron carriers. And I get something else out too, way at the bottom. Many. 1. The only other thing that goes in and it helps break stuff down is we do get a water molecule that goes in. But I'm not too concerned about the water. Does it carry energy like I care about electron carriers? And on your table. I feel like I had more Krebs cycles on the other side. I'm going to leave this up here. It's still got the big version, or a smaller version of that big Krebs cycle on there. So for where does this occur? This is where we get a little bit of variation depending on whether you're a eukaryotic cell or a prokaryotic cell. And so in advanced A&P, you guys only learn all of this for eukaryotic cells because we are eukaryotic. This is microbiology, we talk about prokaryotic cells. So in a eukaryotic cell, so this would be like even in your cells, this is all happening in the matrix of the mitochondria. Fluidy area inside the mitochondria. Eukaryotic cells, matrix of the mitochondria. Is a very advanced organelle. And so prokaryotic cells don't have a mitochondria, but they still do this. And so in prokaryotic cells, this happens in the cytoplasm, happens in the fluid. What goes in? Not a lot goes in. 2 acetyl-CoAs. I only drew one, just know this one goes into the Krebs cycle, from the pyruvic acid that we had another acetyl-CoA, another one would go in. The 2 acetyl-CoAs would go into Krebs cycles. I only drew one, otherwise never have enough room on my board. Water goes in. Great. So what comes out? And again, you have to remember number-wise. I drew up one Krebs cycle, but there's also another one happening down here from this other pyruvic acid that got broken into acetyl-CoA that went into the Krebs cycle. So one thing that comes out is, start up here, carbon dioxide. But if 2 come out in this Krebs cycle and another 2 come out in the other Krebs cycle that I didn't draw, how many total carbon dioxides have come out? 4. So what comes out? 4 carbon dioxides come out in the Krebs cycles. So again, this is all happening from just the start of one single glucose. So 4 CO2s are coming out. How many NADHs? 6 NADHs, 3 here and 3 in the other Krebs cycle I didn't draw. How many FADH2s? 2, 1 here. And how many ATP? 2. 1 here and 1 on the other one I didn't draw. So I get a lot of stuff out. So for energy produced, I get 2 ATP. Still not very much ATP, but I get some. Without anything else important, you can even say refer to the PowerPoint if you don't want to try to draw it in. The biggest thing that we are getting out of the Krebs cycle, for me, it's the anything else important. We are getting a lot of electron carriers. We got 6 NADHs and 2 FADH2s. That's a lot of electron carriers because our next step is all the electron transport chain. So I get a few precursor molecules and all these little arrow steps. The biggest thing is we get a lot of electron carriers. Is it part of fermentation, aerobic or anaerobic respiration? Depends on the organism. If it can do this aerobically for aerobic organisms, and it can do it anaerobically for anaerobic organisms. Again, in A&P, we only focus on us, and we are aerobic, and so we only do this aerobically. But some bacteria are anaerobic and can do this anaerobically. Electron transport chain. I'm going to fill some of this in because I don't know if I'm able to draw it all out. So for where does the electron transport chain occur? Again, this is going to depend on the organism. If it is a eukaryotic organism, this happens on the inner membrane of the mitochondria. The structure of mitochondria, it has like an outer membrane and it has an inner membrane. So this happens on the inner membrane of a mitochondria. But again, bacteria do not have mitochondria, way too fancy for them. But it still happens on a membrane. This all happens out on the cell membrane. So it's still a phospholipid membrane, but it happens on their cell membrane. What goes in? It's the electron transport chain. So all those electron carriers are what goes in. So this is where we have to add up everything we've made so far for our electron carriers. So how many NADHs have I generated between the glycolysis, the making of the acetyl-CoA, and the Krebs cycle? How many NADHs have I made so far? 10. So what goes in? 10 NADHs. How many FADH2s? Just 2. We just got those out of the Krebs cycle. For what comes out, we get ATP. I'll give you the number in that next little box. We get ATP and we get water. Energy, it varies between about 34 to 36 ATP. A lot more than just like 2. We get between 34 and 36. Again, there's a difference because some organisms are more efficient than others. Anything, I'll show a picture really fast. Again, you can just like note. The biggest thing is the electrons eventually have to land somewhere, and they have to find a final electron acceptor. It's generally where we get our water. The biggest thing that happens here is that all the energy in those electrons, I be carried about the electron carriers, all that energy is used to pump hydrogen ions across the membrane. It's by pumping tons of hydrogens across the membrane where they're super concentrated and they don't want to be concentrated. It's called a gradient, how many are on one side compared to another, is eventually used to make the ATP. No, I'm going to skip this slide. So we take the energy that those electrons that are in that NADH, there's some electrons, it's used to pump hydrogen ions over here. So we have a ton of hydrogen ions, they draw 3. They don't like to be crowded, so they want to go across to the other side of the membrane. Well, there is an enzyme embedded in the membrane, whether it's in the inner membrane of the mitochondria or the cell membrane on a prokaryote cell. Those hydrogen ions are like, we don't want to be crowded. No one does. We want to sneak across. And they can go through this enzyme, big blue blob. And this enzyme's called ATP synthase. Again, it's named for what it does. It's an enzyme ACE that makes ATP. When that hydrogen ion goes through this enzyme, it spins the top of this, kind of like a wind turbine. And that energy from the spinning, from that hydrogen going through, is the energy that makes the ATP. You take all the energy in the electrons, pump hydrogens to where they're crowded. As they sneak back, they spin the turbine and generate ATP. I was going to try to draw that, but I don't have time. I got all through all the steps. This is your book picture. That's like horrible looking. This one looks a little more basics of what's going on. But I'm going to end right there. I'm going to tape the rest of this and put it out there because I was going to try to get as far as I can. I knew I wasn't going to quite finish it. I will tape the rest of it, so you guys do have to listen to it. It'll probably be around 20 minutes, maybe 15, 20 minutes, somewhere in there. So finish watching or listening to the rest of it, and then there is the normal quiz that will be on it. Forgot to look at the calendar of like what's all coming up. So I know there'll be a quiz due before I see you on Wednesday, so it'll be a quiz due on this chapter, due tomorrow night by midnight. And then I think the next test is due Wednesday night by midnight. So we're going to start our next PowerPoint. I just don't remember what chapter number it is on Wednesday. And take a quiz by tomorrow night. I think that's the only thing you have to do before I see you on Wednesday. Otherwise, I will see you Wednesday. If you do have any questions, send me an email. Come around. Let me get more colors.